Welcome to this subunit on situ conservation of plant genetic resources. This subunit is part of Unit 1.1, Plant Genetic Resources within Module M1, Biodiversity and Agro-Resources. This session on situ conservation of plant genetic resources. In addition to this video, this subunit includes a link to an article on the vulnerability of situ conserved germplasm. At the end of this course, you will be able to identify the different types of situ conservation of germplasm, to design germplasm conservation techniques for different types of seed, to apply techniques of situ germplasm conservation. The subunit is divided in 9 sections. A first one on ways of situ conservation, a second one on orthodox seeds, a third on recalcitrant seeds, a fourth on intermediate seeds, a fifth on field collections, a sixth on in vitro conservation, a seventh on cryopreservation, an eighth on pollen storage, and a ninth on DNA conservation. The situ conservation of germplasm can take place in different ways, such as seed banks, banks for conservation of vegetatively propagated material, in vivo conservation in field collections, in vitro conservation, using slow growth systems, by cryoconservation, by encapsulation, or in DNA banks. Seed banks conserve seeds, and depending on their behavior to orthodox conservation conditions, low temperature and moisture, they can be classified in orthodox, recalcitrant, or intermediate seeds. Several tests are applied, consisting in desiccation of seeds at different degrees and depending on tolerance. Seeds highly tolerant to temperatures are considered as orthodox. Those that do not resist even moderate desiccation are considered recalcitrant, and those with intermediate behavior are classified as intermediate. It is possible to know which seeds belong to each category by making a search in specific databases, like the one of Kew Gardens. In this slide, we can see the results of cucumber seeds, which are orthodox. Fortunately, many species have an orthodox behavior regarding seed conservation. This means that their viability period can be extended by decreasing moisture content and temperature of storage. The longevity of seeds can be calculated by using Harrington's rule of thumb, which states that seed longevity duplicates for each 1% of reduction of seed moisture, or for each 5 degrees of storage temperature reduction. In addition, both effects are additive. An additional development are the longevity equations of Ellis and Roberts, which state that within certain limits there is a logarithmic inverse relationship between seed longevity and their internal moisture, as well as from temperature. The conservation of orthodox seeds generally takes place by desiccation of moisture levels at levels of around 5%. And depending on the temperature of conservation, there are different collection types. Base collections intended for conservation for very long periods, even hundreds of years, are conserved at -18 degrees centigrade in sealed containers. The active collections, which are for continuous use, are generally conserved at 5 degrees. In addition, there are security duplicates, in which accessions are deposited in black boxes, in which the conserver does not know the content. One example of security collection is the Svalbard seed vault, where seeds are stored at -18 degrees centigrade. The vault has already stored around 890,000 accessions and has a capacity of around 4.5 million, and it uses a black box system. Seed desiccation can be performed using different procedures, such as desiccation chambers or with silica gel. Also, a precise determination of seed longevity can be calculated using seed viability equations, which using different parameters can determine the viability of seeds after a certain period of storage. In a study in maize, it has been found that at 4 degrees centigrade, the viability almost did not decrease after 20 years in storage, but this effect was more pronounced when stored at minus 18 degrees centigrade. As stated before, some species are recalcitrant and cannot be desiccated or stored at low temperatures. Here are some examples of recalcitrant species, such as rubber, mango, cocoa, cocoa, avocado, oak, chestnut, jackfruit, or lychee. On many occasions, recalcitrant seeds are fleshy seeds and therefore do not tolerate desiccation. The conservation of recalcitrant seeds can be performed at a short and medium term. In this conservation, the seeds are conserved at the lowest possible temperature without moisture loss and eliminating mycoflora. However, there is a different sensitivity depending on the species. The long-term conservation can be done through cryopreservation at minus 196 degrees centigrade or at minus 80 degrees centigrade, but this can be done only with small seeds and in very few cases. Long-term conservation of embryos by cryopreservation is also possible, only if they are small and able to rapid dehydration. An alternative to seed or embryo conservation is making in vitro conservation or conservation in field collections. Other species have seeds with an intermediate behavior between orthodox and recalcitrant. In general, conservation of these materials, such as coffee, oil palm, araucaria, or orange seeds, is possible at 15 degrees centigrade and 10% relative humidity. An alternative are field collections, which are technically very simple, as plants just have to be grown in the field or greenhouse. However, field collections have several disadvantages. For example, they require a lot of space and labor. Also, there may be attacks of pests and diseases, and also viruses, viroids, and other pathogens are propagated with the vegetative material. And they may also suffer from climatic disasters. The in vitro conservation may be used for species which are recalcitrant or intermediate seeds, as this is a way to conserve many materials in a small space. For vegetatively reproduced crops in which it is of interest to conserve specific genotypes, or to conserve sterile materials that cannot be propagated by seeds. The in vitro conservation can help in ensuring obtaining disease-free material by using techniques such as meristem culture, micrografting, thermotherapy, or chemotherapy. However, in vitro conservation has several drawbacks. For example, it has a short regeneration period, every few months, although it can be extended by cultivation at reduced temperature, using osmotic agents to reduce availability of water, or using growth retardants like abscisic acid. In vitro conservation also is more expensive than field collections, as they require specialized labor, expensive consumables, climatic chambers, laminar flow cabinets, sterilizers, electricity, etc. And also somaclonal variations resulting from mutations taking place in vitro can appear. Cryopreservation involves bringing the tissues or seeds to a state of zero metabolism due to super low temperatures, minus 196 degrees centigrade in liquid nitrogen or minus 80 degrees centigrade in deep freezer. In these conditions, all biochemical reactions are arrested. The following steps are needed for cryopreservation. First, establishment of in vitro cultures or the sterilization of tissues and organs taken directly from the plant. Second, addition of an appropriate cryoprotectant. Third, subjection of cultures, tissues in this case, to low or super low temperatures. 4. Storage of frozen cultures in liquid nitrogen. 5th, thawing of retrieved cultures. 6th, removal of cryoprotectant by washing. 7th, determination of viability. 8th, reculture of retrieved material. And 9th, induction of growth and regeneration of plants. Freezing is achieved by one of the following three methods. Fast freezing by direct immersion of the material in liquid nitrogen, or pre-freezing the cultures in various steps to -20, -50, -70, -100 degrees centigrade and then -196 degrees centigrade. Or third, gradual and slow freezing at a determined slow rate, 1 to 5 degrees centigrade per minute. Some special techniques, like vitrification, are also involved in cryopreservation. Vitrification consists in the treatment of cultures with high concentrations of cryoprotectants so that at low temperatures the solution forms a metastable glass instead of a crystalline solution. The survival and the extent of regeneration of retrieved cultures subjected to cryopreservation depends on a number of factors, like the physiological state, age, and nature of the culture to be preserved, concentration and nature of the cryoprotectants, method of freezing, storage temperature, method of thawing. A disadvantage of cryopreservation is that good protocols must be available, and these depend on species and genotype, for an adequate storage and regeneration. Here we have some examples of cryopreservation in potato, Solanum tuberosum, and in garlic, Allium sativum. An alternative to seed or vegetative tissue conservation is pollen storage. However, only the masculine gamete is conserved. The advantages of pollen conservation are that many gametes can be conserved in a small space, and there are not so many phytosanitary issues for a change of germplasm as with seeds or vegetative tissues. The disadvantages are that it requires crossing with a maternal individual, the cytoplasmic part of the genome is not conserved, and the longevity of pollen is generally shorter than seeds. DNA conservation can be complementary to the conservation of germplasm. Having DNA conserved can be useful for molecular marker analysis without the need of accessing the germplasm, for studies such as the measurement of genetic diversity, the estimation of genetic relationships among materials within a collection, the identification of duplicates, the development of nuclear collections, the assessment of genetic stability, for phylogenetic studies, or for the development of germplasm conservation plans. In this case, the extracted DNA is conserved at ultra-low temperatures. Users receive the DNA as well as data on passport of the accessions or materials, DNA quality and concentration, extraction protocol, primers and other molecular data, sequence information, related literature references, links to other data from the germplasm bank, and the donor of the sample. In this slide, we can see some DNA banks where DNA is stored. The most important messages of this subunit are: first, germplasm can be stored at situ in different ways, although seed storage is the most common way. Second, seeds can be classified in orthodox, recalcitrant, and intermediate depending on their tolerance to desiccation. Third, orthodox seeds can increase enormously their longevity by reducing their moisture and temperature of storage. Fourth, genetic resources having recalcitrant seeds can be stored in vitro, in field collections, or cryopreserved. Fifth, in vitro conservation and cryopreservation have considerable technical requirements. And sixth, storage of pollen or DNA is an alternative in some cases. Many thanks for your attention.